Okay, good morning, everybody. Okay, so let's get started. We're almost on time. So I'm David Pearce, and I'm speaking really as a scientist and as a board member for the BDSRA today. So many of you have known I've been coming to these meetings as a researcher for far too many years. And we thought after last year it'd be good to have a general overview. I'm not going to be talking about any of my research, but I'm going to be talking about all of the good things that are happening out there with respect to other people's research. But first, I need to remind you that there are many tables outside where there are other researchers and scientists that are recruiting people into studies. A lot of the good things that are happening in Batten disease right now with respect to clinical trials are because of these research studies that are here. So in your bags and your packets, there's information about the different research studies. The researchers are very accessible. Please try to participate in these studies as much as you can for your own children and for the future generations of children with Batten disease. So just by way of background, as I said, I'm not going to be talking about my research, I'm just going to do the overview. So there was a Genetics 101 this morning, which was just a basic introduction. I'm just going to revisit a little bit of that, just because some of you weren't here, just to set the table. So then I'll just talk about the different forms of Batten disease very, very generally, and then we'll get into the really exciting stuff, which I know you want to hear about, which is the therapies that are being developed for these diseases. So just to remind ourselves, what exactly is Batten disease? So Batten disease is this: we have a cell, and there's billions of cells in the body, and they all have something called the lysosome there. The lysosome is the recycling compartment of a cell. Just like anything in life, we need to recycle. So basically the components that make our cells work, they have a finite lifespan within the cells, so they need to be broken down and then rebuilt up. So the lysosome is the compartment that does that. So the scissors in there are really what's breaking up all of these components that get shipped essentially to the lysosome. So, well, what is a lysosomal storage disorder? That's what Batten disease basically is. It's when those scissors break. So if those scissors break, that means that the cells are unable to recycle. If they're unable to recycle, then we're unable to probably make other components of the cells. So in essence, the lysosome becomes full of garbage. Now, biology is very complicated, and there's been many studies trying to understand the basis of this, and we're still trying to figure out some of this. What is confusing to us is there's at least 12 forms of Batten disease, and the proteins that are involved in this process all have different functions. And I'm going to go through those very, very briefly, just so each of you know the different forms of Batten disease that we're talking about. So, okay. Remember, I don't practice these talks anymore, I don't have time. So just before we get to there, transcription and translation, all of the gene products that we're going to be talking about. So you hear about CLNs. So just to review there, because some of you get confused, I know, with CLN and what PPT1 is, TPP1. We have a lot of nomenclature, and a lot of jargon there. Hopefully Helmuth did a good job of running through some of that this morning. But what exactly makes these components in the cell? So what we actually have is what we call transcription and translation. So you all hear this word mutation, okay? We are all made up in this room of 3.2 billion base pairs. That's our genetic code, 3.2 billion letters. If I was to stand here and recite that, it would take me 9.5 years to recite that. That's how long it is, right? It's that one lecture that you hated in college. So transcription and translation is this. So that code of life is then imprinted into something that's the DNA, goes to RNA, and then makes the protein. So when I talk about the different genes, we have the CLNs, that's the gene, and then we'll talk about the product, which is the protein. That's the nuts and bolts. That 3.2 billion base pairs makes about 21,000 nuts and bolts in the cell, and that's what makes the cells work. So CLN1 makes something called PPT1. That's basically, it's an enzyme that sits in that lysosome. So it's like a pair of scissors that we talked about. What that pair of scissors does is actually removes a piece of fat that attaches to proteins that makes them work. So if that PPT1 is not working because it has a mutation in CLN1, those scissors are broken, and then you get accumulation of garbage. CLN2 codes for a protease, which basically degrades proteins in the lysosome. So it's an enzyme, again, that sits in the lysosome, so it literally is a pair of scissors. And if you have a mutation in CLN2, means the TPP1 protein is not functional, and those scissors are broken, and you get accumulation in the lysosome. CLN3 sits on the outside of the lysosome for the best we can tell, and we honestly, if anybody tells you they know what it does, I would probe them with a lot of questions because we really have no idea what it does. We do know that lots of disturbances in the lysosome occur if you don't have this particular protein. And some of the therapeutic approaches that I'll talk about are targeted with respect to that. CLN4 is for one of the adult Cuffs forms of Batten disease. And really, this is a protein that we think is involved in maybe chaperone or folding or helping deliver the types of proteins that go to the lysosome. So again, if there's something going wrong with delivery to the lysosome, that's what may be causing the accumulation or the garbage accumulating in the lysosome. CLN5, again, is another one that we don't really know too much about. I'm going to go through all of them because I know some of you have these forms, and I can't just ignore them. So I'm sorry that I'm saying we don't know much about what they do. It's just what it is. So CLN5, there was a publication that came out recently. We think it's involved in delivering things potentially to the lysosome. So if things aren't delivered appropriately, then you get accumulation. CLN6 is much the same with respect to that. CLN7 is interesting. It actually, much like CLN3, it resides in the actual outside of the lysosome, and it's basically, from what we can understand, it transports components into and out of the cell, into and out of the lysosome. So that tells us that the environment of the lysosome is very important, but we don't know exactly what it transports. CLN8, again, is involved potentially in things that are delivered to the lysosome. CLN10 is another pair of scissors. CLN11 is another one that's involved potentially in delivering things to the lysosome. And CLN12 is another protein that we think is involved in transportation of some sort of components into the lysosome. What I think you just need to understand just from this is that it's kind of complicated. And don't think that we're all super smart scientists, because if we can't figure it out, we've got a long way to go. So ask your questions, because it's really very important. The point being is the lysosome is the garbage disposal. It's this exquisite piece of the cell, and changes in multiple parts of the cell can disrupt this process. And unfortunately, this part of the cell is very important for the brain and neurons in the brain, and that's why Batten disease has these terrible progression in the neurological consequences. All of these processes are very important for the understanding of how brain cells actually work. So, thanks to Dr. Jill Weimer, because I stole her slides for some of this presentation, and if I didn't acknowledge her, she would be really curmudgeon-y about it and go running around telling you all that I stole her slides. So as many of you know, Jill is really, from the Stanford side of things now, the leader in Batten disease research. And she's really gone on a tear in terms of understanding all the different therapeutic approaches. So I just told you it's complicated, right? So why would we focus on one way to try and cure these horrible diseases? Let's throw the kitchen sink at this approach. So what I'm going to do is go through the different approaches and how far along other individuals in this room actually are with these therapeutic approaches right now. What I need to tell you first of all is I will be talking about clinical trials. So there are clinical trials that are ongoing, and there are individuals that are very close to having clinical trials. A clinical trial is the testing of a new treatment for a disease. This happens across all areas, whether it's in oncology or cardiology, but obviously we're going to be talking about Batten disease today. What you need to know is a clinical trial is a highly regulated process. You often hear about the boogeyman, which is the FDA, the Food and Drug Administration. Don't let anybody tell you they are the boogeyman. The FDA has been tremendously good to work with for us in this field. They do recognize that Batten disease is a devastating disorder. They don't necessarily tolerate placebos, where we have to give a child no treatment to compare to treatment. They understand that we can have to try and treat these diseases in an open way. They've been very open with respect to that. The other thing which I think is very important for you to understand, conceptually a clinical trial in most instances, so for example in cancer right now, if you're recruited into a clinical trial, that's an increased treatment option. It's on top of standard of care. So one of the things that we will obviously have to move with Batten disease is the fact we'll have many treatment options out there. So I ask all of you that are developing these types of approaches to think about that. We have a very small pool of individuals with these diseases. We need to have increased treatment options that are defined as clinical trials. And where did I put that? Okay. Okay, so first of all, gene therapy. I'm not going to get into the technology of all of this for two reasons. One, because I don't really do that. And two, I only have 45 minutes to overview. So what I thought I would do is just give you some of the jargon that becomes very complicated. I think it was not at the last NCR meeting, the one before that in Boston, we had all these gene therapy presentations, and everybody bamboozled everybody with their approach for gene therapy. What's important to know is whatever a therapy is, you have to deliver it. The key thing for a therapy is to get it to the site where you want to treat, and obviously in band disease, it's the brain. The other thing is how does it actually work? So the jargon I just want to go through for gene therapy is you'll hear intrathecal, you'll hear intravenous, you'll hear intracranial. So I'm just going to walk you through what that actually means. And then we'll talk about the different trials very, very briefly, what people are doing. And then you go and ask the people who are developing these things, well, what method of delivery are they doing? My apologies to all of you who are developing these. This is going to be a very high-level talk, and you're going to hate it, okay? Because I'm not getting into the technicalities, and you're going to say, well, you should have said this, you should have said that. Okay, this is just a high-level thing. Okay, so what is intrathecal? So intrathecal is this, and please bear in mind right now I can't see what I'm doing here. So, okay, intrathecal gene therapy is this. Okay, we want to take the CLN3 gene. So what exactly does that mean? Gene therapy is this. It's simply taking a virus, not too unlike a cold virus, taking all the snotty stuff out of it, and then putting in the genetic material that you think is important. So if you're missing the CLN1 gene or have a defect in CLN1 or a defect in CLN2, the concept is you put the gene back into that virus, you put it back into the person, and hopefully that spreads throughout everywhere that you want it and replaces the broken copy using the virus. Now, of course, we can clear a virus through our immune system, so it's not as simple as that. Okay, so we take the CLN3 gene in a virus, and it's not quite as simple as that. It's a lot of regulation, and it has to be highly purified because you're going to put it into a human being. And then you do this after you've done a huge amount of preclinical studies. So you've tested this in the mouse, you show it's safe, and you show it has efficacy in terms of potentially curing or treating the disease in the mouse. So, and every trial that's ongoing and every trial that's under development is classified to start with as a safety trial. Your children are precious. If this is done for the first time, it's first do no harm. And that can be frustrating sometimes, but that's the rules that we have to abide by. That's the ethics that we have to abide by. So, so intrathecal is this, and my apologies, it should actually, the syringe should be a little bit higher. It's basically a lumbar injection into the spine, so that then the virus, through the cerebral spinal fluid that populates the spine and goes into a part of the brain called the ventricle, can then spread the virus throughout the brain. So the target being the brain. So intrathecal is that type of delivery. Intravenous is the same concept. You could take the same type of virus and then inject it into the bloodstream, and in theory it should go throughout the whole of the body. Intracranial is a direct injection into the brain. And I filled the whole brain there but the problem being there is in a mouse, that's what can actually happen with a few injections, but the mouse brain is the size of my thumbnail. Obviously, we're dealing with something much larger here, so intracranial injections are probably much more targeted to small brain parts of the brain right now. So just to pause, that's the different ways that it can be delivered, and I'm going to talk about the ongoing trials that are under development and in process right now, and I'll call out the individuals that are associated to those, and they can get into more details with you. It's not for me to do that. What is also very important is if you think about, well, we can deliver a virus and we can deliver a gene, but how much comes off of that virus? So, because many of you will be saying, well, why are so many people doing this? And why are people essentially in competition for this? There's different ways to get the virus to make 5 times or 10 times or 20 times the amount of the product that you actually want. So the devil in the detail is we don't know how much we need to get in there to be really corrective. So these different vectors and different viruses have different levels that come off of them. Some of these viruses also go to different cell types in the brain. The brain is of billions of cells. So some of these vectors that are being developed, or viruses that are being developed, have a better or worse ability to populate the whole brain or different cells. That's very important. That would be, if I got into all the details, that would be like a 3-hour lecture, and trust me, you don't want to hear that, because those in the audience who are more sophisticated than me wouldn't want me to do that lecture anyway. So where are we at? So clinicaltrials.gov. This is the place that you always check for clinical trials. Right now, if you run a search for Batten disease at clinicaltrials.gov, you'll get 30 hits, okay? But what's important to look is it tells you whether it's open, whether it's accruing or not. Many of these are studies where they're just following the children for natural history studies, for example, and that should be up there. If you're looking for therapeutic trials, you just have to take a careful look. And then there's a couple up there that have been closed for recruitment, and that. So, but what I will tell you, and I'll just boil it down for you right now, is in terms of true therapeutic trials that are actually open, there's 2 for gene therapy. For CLN6 and CLN3, and they're being run by Amicus at Nationwide Children's Hospital. So anybody from Amicus, if you'd like to stand up, and these are the individuals that you would go and talk to. Okay, there we go. And thank you, because these are the only 2 gene therapy trials open. There's been a huge amount of work to get these trials actually open, and they've gone through all of the regulatory components right now with respect to that. So also under development, Regenx Bio are also developing a gene therapy clinical trial. Regenx, would you like to stand up? Okay, these are the people you need to talk to, remember. And then Abeona have one under development. Actually, Abeona have got FDA approval for a CLN1 clinical trial. I don't believe Spark Therapeutics are here. Okay, they are developing a gene therapy, and we already did Amicus. So anyway, so the thing is, trust me, I've been doing this for many years, and the fact we have open clinical trials is a tremendous milestone. Now, when you go and talk to these people, remember, these are highly regulated. These are safety trials. Some children will be included, and some children cannot be included because of the profile of safety to start with and efficacy. So, and trust me, all of the individuals in the design of these trials are very thoughtful individuals and care very much for the children. And they will explain those criteria. Once we get through those first safety trials, then we try and get every child in there. And trust me, I'll be the first in line to come and help you beat these guys up to get every child in these trials. Okay, so next up, what have we got here? Enzyme replacement. So this is the huge success story that we have in Batten disease. And you'll hear from many people in the industry that Batten disease is really the trendsetter with respect to clinical trials. But we already have one treatment for Batten disease, and it's called Brineura. It's enzyme replacement therapy. So basically, it's the idea if we can get that pair of scissors back into the lysosome, it clears the storage material. So what these individuals at BioMarin have done over a number of years is they've actually used a way to manufacture that TPP1. They've manufactured the CLN2 product because it's a soluble product. It means you can literally just make it and stick it in water and dissolve it, and off it goes. Except we don't want to put it in a glass of water, we want to put it in the brain. So they've developed a specific technique now, so every 2 weeks children can receive Brineura through a port. It delivers to that reservoir of cerebral spinal fluid, and then it can populate the brain. So they should be congratulated, and obviously some of you, if you need to ask more people about, questions about that, there's many parents who have children here on this treatment, so I encourage you to speak to them, but also there's some people from BioMarin in the audience. If you could stand up. So thank you for that. So then one other group that's actually here is in what we still call the preclinical stage, just like I talked about under development for gene therapy. Collaborations Pharmaceuticals are here, and they're essentially trying to recapitulate what BioMarin have done with PPT1 or CLN1. So they're developing a process to manufacture PPT1 enzymes. So Sean and gang from Collaborations, are you in the room? Okay, next up, stem cell therapy. We don't hear so much about this now. A number of years ago, we used to hear a lot about stem cells. And there was actually one clinical trial where 6 children with Batten disease were treated really with fetal-derived stem cells were injected into 6 portions of the brain a number of years ago. And they turned out to be safe. Unfortunately, there was really no efficacy shown with respect to this transplantation of stem cells. And I think with the way things are evolving right now in science, there's a lot less investigations being done with stem cells right now. But I thought it made sense to point that out, because I know many of you will ask about stem cells. There are no ongoing clinical trials or preclinical studies with stem cells that I'm aware of right now in the therapeutic realm. So anti-inflammatory compounds. A lot of you here, when John and his lab get up here, and possibly Jill, they'll talk a lot about astrocytes and microglia and things. So these are the cells, the support cells in the brain that help support the neurons and make sure they're healthy and functional. And an indication of a problem in the brain, whether it's traumatic brain injury or whether it's something like Parkinson's disease or Batten's disease, is the degeneration of the brain going on. So these are the types of cells that seem to become activated. And we could probably have a very long debate with a lot of the scientists in terms of what their contribution is to the inflammation or trying to ablate the inflammation is right now. But we do know trying to target those cells makes a lot of sense. So one approach is, and individuals have a lot of science, now we're getting much more down to the bench science, trying to find ways if we can actually target inflammatory responses. One of those, which was a spin-off from that, is we do know there's an autoimmune component in the juvenile Batten disease, and our colleagues from Rochester completed a safety trial with respect to mycophenolate, which is Cellcept, which was to dampen down that autoimmune response. I think the jury's out, really, John, on that. Yes, I mean, it was safe, but I think other therapeutic approaches like gene therapy and that have really sort of overtaken that type of approach right now. But nevertheless, it's important to sort of, to realize that many people are looking at ways to use small molecules. And when I say small molecules, a way to deliver a pill that actually have an impact on it. Thus far, everything I've talked about is much more complicated in that delivery realm that I've talked about, whether it's gene therapy, I've talked about injections, I've talked about enzyme replacement. These are quite complicated procedures. Some small molecules could be delivered orally. They could be introduced into the bloodstream. So you'll hear a lot about these types of things, and I'm sure there'll be some posters at the session there. So other small molecule therapies often focus on the fact that Batten disease, we know, is a unique set of diseases, but there are many attributes we know with respect to the brain being affected. So obviously there are several other brain diseases, whether it's Alzheimer's or Parkinson's and that. So many drugs that are being developed to try and slow down what's happening in those diseases could have application to Batten disease. So, and some of these may have modulating activities in terms of functioning in the brain, but the ones that we try to focus on really are ones that we think that could change the activity of CLN1 or compensate, CLN proteins. So we know that the CLNs are missing, so can we put a drug in there that will bypass that? Or if there's a small amount of that CLN product still there, can we get more of it? Also, we want to try and clear the storage material. Is there a way that we can clear that storage material? So these are drugs there that, and there are ongoing studies, preclinical studies. So Polarex, I don't know if the Polarex are in the room. So they have a compound that really sort of does two things. It potentially can help clear the storage material and then elevate the activity of some of the CLNs. Circumvent, are you in the room? So Devon? So they have a molecule that basically, and you're really going to hate how I describe this, smashes up the storage material. Okay. So the point being is, and these are all preclinical studies yet again, and then the final one I'd like to talk about is Greg or Danielle in the room from Beyond Batten? So many of you are familiar with Craig, and he's very close to submitting to the FDA for a clinical trial using a combination of a couple of drugs that will help bring up the activities of the CLN pathway as well as help clear the storage material. Say? Yeah. So, so there's, and again, that will actually be, can I say how the delivery is? Is that okay? It's orally and intravenous into the blood, so a nice simple type of treatment with respect to that. So there's a lot going on in that space. And I think it's time to pause there and say, okay, we've got all these things happening. Everybody that I've talked about, I've spoken to, we're all aware that combinations are going to be very important. Gene therapy specifically targets the DNA and corrects that genetic defect, and that can help cure the disease. But we've already had the disease there for a period of time. So these other approaches, whether it's clearing the storage material, whether it's smashing up the storage material, whether it's actually trying to induce the pathway, is all going to culminate in a better treatment for Batten disease. So where are we at next? So lysosomal modulators. I talked a little bit, and this is most relevant to CLN3 and some of the others that we don't know what the proteins do, is that there's a lot of work that's being done. We know the lysosome is messed up. It may not be a specific pair of scissors that's broken, but there's lots of work out there that shows that what we call the homeostasis, so the happiness of the lysosome, is disrupted in some way, whether it's the pH or whether it's calcium. Calcium and potassium levels have become a very hot area to be studied in Batten disease right now, particularly in CLN3, but also in others. So if you think about this, what that would mean is if you take a glass of water and you taste it, it's okay. If you put salt in there, you kind of spit it out, right? So the homeostasis of the lysosome is this: if you change the environment, it goes bad. You don't like it. And enzymes and scissors are just the same. So if something like calcium or the acidity of the lysosome is changed, and those scissors will break or not work effectively. So can we modulate that in some way? So there are many people screening drugs, again, that can we deliver those in a variety of ways that would restore the balance or the homeostasis or the happiness of the lysosome. So natural therapies, I thought it's important that we have this one in here. So we've known anecdotally for many years that giving our children carnitine or fish oil certainly seems to have a calming effect with respect to some of the symptoms. And we shouldn't turn a blind eye to that. It's clearly one of those combinations that we should include. It certainly doesn't do any harm, and I suspect there will be other compounds that will surface as we explore them more and more. So then finally, RNA DNA modulation one. So we're going to finish on more of the geeky science because this one's really hard to explain in lay terms. So if we go back to the fact that we have DNA, and if you have Batten disease, you have mutations or changes in that sequence. Okay, it's broken in some way. And that means when you go to make the protein, but you go to make RNA, which messages to make the protein, it's broken. It doesn't code. The code is incorrect. So how do we fix that code? I mean, gene therapy is a complete replacement of the entire thing. We know that with some of the mutations, there is something that's called a stop. So as we read through the code here, we have something we read through, because think of it, you've got 3.2 billion base pairs. We don't read the whole 3.2 billion, we just make 21,000 components of that. So we have a start and we have a stop. One of those changes sometimes is introducing a stop in the wrong place, and that's what breaks a CLN protein sometimes. Okay, that's the start and the stop. And that was so there's basically the blue pieces there are when we make the protein, whether it's PPT1 or PPT2. So then a stop, and the red is the stop. I mean, I'm not going to get into the coding here. The green is go, red is stop. So if you have a change in your CLN and you have a stop there, rather than make that entire chain of blue, you only make a portion of the blue. Okay? So then a portion of the blue means it's not functioning. You've only got a fraction of the protein there, a fraction of PPT1, a fraction of TP1, a fraction of CLN3, whichever one it is. So the geeky science here is if we dump onto cells something that means ignore the stop, a compound, and there are compounds being developed right now, and they're being tested in other diseases such as cystic fibrosis and muscular dystrophy, because they have a lot of these stops. Can we jump on and take these drugs and then read through the changes that actually happen in CLN proteins? So that's all of the different approaches that are ongoing right now. So hopefully that's been a good overview, because I know you guys crave for information. The one other disclaimer is there's lots of other work being done. All of the companies I talked about, we had present to the board on Thursday. Your participation in these trials and these studies is obviously going to be very important. There are many other people screening lots of compounds and doing this, and I am not privy to everything. I know what most people are doing, but not privy to it all. So this wasn't everything that was being done. So with that said, that was the high-level overview. You know who else you can talk to now with respect to the different trials and what they're developing. What I thought knowing this crowd would be is that there'd probably be several questions, and I'm allowing time for that now. Thank you. Yeah.
Most of these therapies right now can either slow down or stop the progression of disease. There's nothing really to really reverse it, but if the combination therapies that also focus on removing the storage material that's already built up, will that restore function to some of those cells, or are they already?
Okay, boy, there's a lot in that question there. Right, okay, so I'm going to break it into two, and if I miss something, let me. So I'm going to do, the first piece is most of the therapies are targeted to slowing or halting the disease. And then the second piece is we're using other things that could maybe clear the existing disease. So what does that, and I'm going to paraphrase here, what does that mean in terms of can we go backwards with the disease or go forwards with the disease and bring it back in time? Can we correct, not just stop the disease, but can we restore vision, or can we restore cognitive function or something like that? So I'm going to give you a very sort of general hope answer to start with this. Everything I've described has never been done before. So, and this is the horrible analogy that I use to people right now. If our boys in Iraq have a traumatic brain injury, they can be retrained to gain some of the function, okay? So the terrible analogy is this: bad disease is a traumatic brain injury in some sense of the word. So if you stop the disease process, nobody knows what the consequence to the brain will be. Can it rewire, can it relearn, or whatever? The likelihood of massive regaining of function is pretty unlikely. But the younger the children, the more impact you're going to have. We know from the studies in the mice, excuse me, the earlier you intervene in the animal models, the more restoration or rescue of function that you have. So conceptually in young children right now, and we know anecdotally from those children that have been in the first gene therapy trial, those that are younger and earlier in the stage of the disease are clearly doing better. But we've only been doing this for a very short period of time, okay? So if it takes 5 years to develop a disease, how long does it take to regress a disease? We don't know the answer to that. So I'm going to really, and I'm a little bit outside my realm of expertise. Did I cross any lines there, John, do you think, with respect to, I'm simplifying. I know John's the pediatric neurologist and much more qualified to answer that piece of the question, I think.
Yeah, I don't have anything else to say. It's complicated.
Yeah.
And it's going to take a while to get it through.
Yeah. I don't want you to go away thinking it's a possibility, but we just don't know. So then the second piece is targeting things like storage material. So all of those screens I talked about with respect to clearing storage material, whether it's, if you're doing it in CLN3, it could be directly applicable to CLN2, CLN7, CLN8. We do know from studies that clearing the storage material would be good. So if we can correct the genetic defect, or we can and clear the storage material, or if you can clear the storage material and correct the genetic defect, that's two approaches. These are very complicated diseases. The more approaches that you, more therapeutic interventions, the better, because not one single approach right now addresses every component of the disease. Okay, is that everything? Okay. Yeah.
On your last slide, the truncated protein, are those the things that are being stuck?
So that's a great question. I'm going to have to explain that one to people, I think. Okay, so the idea being is if we read through this genetic sequence and then we get a stop, so we only get a portion of the enzyme or a portion of the protein, what happens to that? Okay, so in CLN1 and 2, we know there's a little bit of activity, just a little bit. Some of the children will have maybe 1 or 2% of the activity of the enzyme. So, but the cell recognizes that this isn't the regular protein it should be there, and it's either broken down, because it's not really effective. In CLN3, we have the truncation. Basically, we know there's about 1/3 of the protein is manufactured in the most common mutations. And this is a little bit of a hot topic with the researchers in what actually happens. But again, it's a piece of junk. It gets degraded down in the cells. And as far as we can tell, there's really not much retention of function of that. So the concept is if you have a smaller piece of activity and you can read through, you can boost that activity up. So that's what the read through is actually about. Does that answer your question? Okay. Any more? Okay, Trace, I keep looking at you, dude. I mean, come on.
We heard a lot about treating the brain disease. Where are we as far as treating the eye disease?
Yeah, so when it comes to gene therapy, and I've got to be very careful here in terms of, there's different types of delivery. To my knowledge, when you deliver intrathecally, the virus doesn't really get to the brain, to the eye, Jill? Some gets them. Okay. So, I mean, the point being, I think, is the eye is a unique, separate piece of the body, right? So I think, we have to address specific delivery to the eye for that. When it comes to, and it's compromised, or it's compromised in different ways. Same with drugs as well in terms of the delivery sometimes. So, and I can't really comment on any studies that I'm aware of with respect to addressing the vision defects. I know it's very important to many of you right now, but I suspect we'll have to have a separate delivery of whatever the drug is, whether it's a small molecule, whether it's enzyme. As far as I know, and I don't know if people who from BioMarin want to comment on Brineura with respect to the eye, that's up to you guys if you want to comment on that. But my general understanding is any drug, whether it's a virus, a stem cell, protein, or small molecule, getting it to the eye is a unique challenge right now. Yeah. 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 That's a great qualification. So what Jill is saying is, and it was actually some of her work many years ago, is that some of us believe the loss of the vision is because what's actually dying in the brain, first of all, is the lack of communication with the eye, and the eye becomes isolated in that sense, and that causes that. Now, it's really hard because the mouse sucks for eye studies, okay? The mouse eye is not anything like the human eye. And you think about it, mice run around the field. Their heightened senses, hearing and touch and things like that. Their vision is not particularly good in the first place. So, yes. So I think to take your question, I'm going to rephrase the question is obviously some forms of Batten disease don't have vision loss, and some forms of Batten disease that are known to have vision loss, some of the mutations or changes in the DNA result in a different course of the disease. And that just gets to us, our poor understanding of it right now. Batten disease is a classification based on the fact that there's storage material. So pathology, that basically, as your child is developing the disease, different tests are done, and it's seen that this garbage is present in the lysosome. That's what the classification of Batten disease actually is, and that's why we've got all these new forms appearing right now. In terms of understanding what it means for each individual Batten disease, that's the bit where I made all these apologies. We just don't know and understand. So the crux of it in your instance, in CLN2, for example, we have what we call genotype to phenotype. So when I just said some people have a little bit of enzyme, and some people have no enzyme, and some people have more enzyme. If you have more enzyme, the likelihood of the course of the disease being slower is more likely. The other thing is that we have 3.2 billion other base pairs influencing this whole process, and we know what 1% of those do. And the final piece there is every child is unique and every child is different, okay? So every child will function very different. Even within certain families who have more than one child sometimes with the same disease, they don't all progress in the same way. So that's really, you know, we've got a lot to learn. Okay, I think John's getting fidgety here. So don't go away. He has some important updates for you on what's actually happening next.